Merci beaucoup, Cissé, mais je tiens à le remercier personnellement pour l'invitation et toute l'association RECONNECTT pour cette invitation, pour être avec vous aujourd'hui. Je suis contente d'être physiquement avec vous, mais d'avoir aussi suivi les différents talks qui ont précédé. Je vais probablement présenter en anglais parce que c'est un peu l'habitude et je serai beaucoup plus fluide quand je parle science. Donc, je m'appelle Emna Harigua, je suis bioinformaticienne. Et aussi, du coup, data scientist, et je suis à l'Institut Pasteur de Tunis. J'ai fait un parcours où j'étais à un certain moment à l'Institut Pasteur à Paris pour ma thèse, puis je suis retournée en Tunisie, notamment pour implémenter certaines technologies où la Tunisie reste un peu en arrière, on va dire. Elle ne suit pas réellement le rythme mondial en bioinformatique dans certains cadres, et aujourd'hui, on espère apporter aussi toutes. Voilà, donc ça va vite. Donc, comme j'ai dit, so as I said, I'm a bioinformatician, and bioinformatics, by definition, is the analysis of biological data that comes in very different formats, sources, and objectives. And it's a very multidisciplinary field, so we can see bioinformaticians that have different backgrounds and then become bioinformatician while they're doing research. So the definition itself of bioinformatics makes it the ancestor of data science in the biomedical field. And this is why AI has to be there with bioinformatics in order to advance the whole field of biomedical research. So my field of expertise is drug discovery. So we work on identifying novel molecules that can be later on developed as drugs against different types of infectious diseases. The therapeutic drug pipeline is a very long and complex pipeline that comes from the discovery, the study of a molecular target within a pathogen, up to the different phases of validation prior to coming to the clinical phases, which are the latest form of validation towards saying this can be a drug administered to people to cure one disease. So this pipeline would take more than 12 to 15 years in order to get one molecule to be validated, and also costs more than 15 to 20 billions dollars for one molecule to reach the market. And the idea of bringing computational technologies, what we call computer-aided drug discovery, into the pipeline is mainly about shortening the translation gap, making the process shorter and also less costly, which is very interesting, mainly for diseases that lack of resources and for countries that are fighting diseases that lack resources. So computer-aided drug discovery, we had different ways of approaching the things. We have what we call, does it work? So we have what we call structure-based drug discovery, where we will be targeting specific targets within the disease. These are mainly proteins. I will be given a concrete example to make things a little bit clearer. And we have what we call ligand-based drug discovery, LBDD, where we are more focused on chemical structures of molecules that should be or can be novel drugs to treat diseases. Then we have a lot of whole pipelines that are done by computer people, such as myself, then go to experiments towards approval. So just to give you a short example, so this is just to say that we as Tunisia, from the African continent, and also having a lot of public health issues that are related to poverty, we have the big challenge of working on neglected diseases of poverty. So for these diseases in particular, which are not very prevalent in the developed countries, we have too little data available to work on, and everyone working on the deep learning approaches understand what is the issue with that. So the data is not abundant, but we also have very heterogeneous data. It is not in good standards towards AI applications. We have also a lot of sparsity and heterogeneity within the data that is published in the scientific papers. So most of the data would come with protocols and approaches that are specific to one group working on a specific disease and so on and so forth, which makes things very, very complicated to bring this data to leverage AI to work on. And also the issue with drug discovery for neglected diseases is also there is no big interest from the big pharmas to develop real medications to treat these diseases, unfortunately, because there is no real market behind them. So, so to give an example on the more classical way of the discovering diseases using computer-aided drug discovery, we will be focusing on proteins. As you see here, this is the example of the main protease of SARS-CoV-2, where we will be focusing on the specific target in order to develop a whole pipeline. So you see the the first three steps of the pipeline are the computational pipeline that we have been developing with our partners in order to first analyze all the data that exists on this protease of SARS-CoV-2, also see what was happening with other coronaviruses related to the SARS-CoV-2 virus, and then set up a whole pipeline of virtual screening where we screen thousands, as you see, 600,000 of docking poses of molecules that then need to be selected. And here we are talking decision making. And most of the time we need machine learning or algorithm that will help us make these decisions. So this is like a high throughput data analysis that also need AI to be implemented. So this is a very classical way of using AI in the process. Afterwards, we go into the blue part. سامحني تنجم تحطهم en mode plein écran. Merci. So afterwards, we will be switching to the latest part where we will be given these molecules. So we went up from 600,000 molecules into only 250, and out of which we were able to validate 2 molecules that were really inhibiting the viral growth of the SARS-CoV-2 viruses. And this work was incredibly done within less than 18 months. With more than 6 teams, international teams working on in order to deliver these 2 molecules for early drug discovery development. So that was the classical way. Now I'll be more presenting what we are currently doing and implementing at Institut Pasteur of Tunis, which is basically ligand-based drug discovery. So ligand-based drug discovery will not be focusing on a given protease, but will be approaching things from another angle, where we'll be screening all the data that exists about molecules that have an interesting bioactivity against a certain pathogen, and learn out of their chemical structures, as our hypothesis is to say, if 2 molecules would be very chemically similar, they probably would have similar chemical biological activity as well. So this is why we will be encoding chemical structures of molecule into binary or numerical data vectors in order to be able to make machine learning algorithm learn out of them, or use graph-based convolution networks in order to encode these structures as we would be looking at our chemical molecules as undirected graphs. And then get some neural networks, learn different features, and be able to predict whether a molecule has the potential to be a real effector against a disease, or generate new molecules that would have similar effects. So that's the pipeline, and it always starts with the data. And when we are focusing on neglected diseases, the issue of data is a real problem. So we started with that in a frame of a project called AIR2D2 that has been financed by the USAID and the National Academy of Science of the United States. We were able to go and look at what exists in the literature, all the data that is there and that is not suitable and not accessible automatically, but we have been working on using different parsing tools of these publications, using image processing tools in order to extract chemical structures. Then we had a lot of manual curation aspects in order to annotate these chemical structures within their chemical activities. We started with data on Leishmania parasite, which causes leishmaniasis, itch and neglected diseases. All the way where we were working on that, COVID-19 hits. So we switched also our work to focus on coronaviruses and anti-coronavirus molecules. And the idea from there is, besides what exists in databases like PubChem, we can have bioassays that consist of hundreds or many thousands of data points. We will be enriching the data from data that exists in the literature and that has higher quality than the bioassays. We stored all this in what we call CDLDB. It's a database that will be soon be released with a publication along that will enable the whole scientific community to access to the data that have been collected by our team, but also will have an embedded machine learning and deep learning tool that will have them predict activities of their own data using the models that we have been training. So the models are different models that we have been working on, and we have been seeing very different performances of what an algorithm, one given algorithm, would render as a performance given a certain data that is trained on. So it's not only dependent on the pathogen, it is also dependent on the source of the data, so the structure of the data and the size of the data. So I'll be presenting one case study. This is the case study where we have been looking on to only data that have been published within like few months after the COVID-19 hit. We have been able to have 2,600 molecules that were manually curated for their effects against different coronaviruses. So it's not just SARS-CoV-2, as at that point we had very little information about that virus, but we have been looking at data on the SARS-CoV that have been hitting in 2003. So what we have is a small data set for deep learning algorithms, and the idea is to say, will we be able to use deep learning algorithm to learn on such a small data set? So we have the size, but we also have a high heterogeneity of the data. It comes from different sources. Some of them are evaluation against one protein activity. Some are viral growth. Sometimes we don't have the information, and so on and so forth. So what we started to do is encoding the data. We use two different approaches: molecular fingerprints. These are algorithms that will kind of cut up the structure of a given molecule and generate a binary vector that would say if a given substructure, as you see in different colors, would exist or not in this molecule. So it's one way or another saying what exists in that chemical molecule in terms of substructures. Then we have the graph convolution approaches that are more suitable for deep neural network applications. Then we have been training different models. We had three machine learning algorithms. We had 4 ensemble learning algorithms, and we have 4 deep learning algorithms out there. And we have been splitting the data in different ways. We have been also doing data augmentation, data oversampling, data undersampling, splitting according to the source of the data, and so on and so forth, in order to see what kind of bias is there. So I'm not presenting the whole things that have been done, but I'll be more straightforward about what we discovered. We were able to see that we have two algorithms, the DAG algorithm and the GCN, that were the best performance among the deep learning algorithm, but so we're not very surprised. The data set is too small, and it was okay. But we continued the work as it's not about just saying we have some kind of performance. We had to make sure that that performance is translated in terms of predictions into something that is intelligible. So we will continue the work, and so this is the optimization of the different algorithms, hyperparameter tunings, and so on. And we were looking at different metrics, so making sure that since the data is imbalanced as well, the idea is to make sure that we don't have issues with predicting one class over the other one, so on and so forth. Still, random forest is better, but it's okay. So what we did after that is that we said, okay, so we have like merely good performances, but what about if we train a model on a subset of the data set, which is already small, and see if it can predict things from another class of that data set. So if I'm training my model on molecules that affect SARS-CoV virus, will I be able to predict the activity against the SARS-CoV-2 virus? So this is an issue that is more important than having 98% accuracy in the domain of drug discovery, because we mainly have data that is too small or not exactly what type of prediction we want to make. So in which case, we will leverage AI in order to learn on things, but be able to predict new things, like training on one task and predicting on another task. And this is what we saw. We saw that GCN was the best algorithm that have a generalization power. So still, it doesn't have the better accuracy or IUC-ROC score, but it was the best one to generalize from one task to another, from one data set to another. We also did a final external validation step. So this is not cross-validation or any type of computational validation of the algorithms. We took a whole different data set that have been done by a different group under other circumstances. And we made sure that there were no molecules between our data set on which we have trained the algorithm and this new data set, which is the FDA approved drugs. So these 1,518 drugs were really tested on the virus growth of SARS-CoV-2. And it is a totally independent data set from our data set. And what we saw, so from 1,500, they only found 17 molecules that demonstrated more or less an inhibitory effect on the viral growth. Using our algorithm, we were able to see that GCN were able to identify 8 true positives out of the 17. So we are here at 50% of a good rate of prediction. So our true positive rate is an important one. If we look at all the molecules, then we said, okay, most of the time when we have selections, we can like score the molecules using a confidence score, probability of being active or inactive. So most of the time we are not looking to the whole set. We are classifying and making scoring things. So if we would take 80% confidence thresholds, we will be identifying 5 of these molecules out of the 17. So this is what brings up 8 to 5 and breaks the true positive rate down to 40%. Knowing that in drug discovery, the most of what we see from virtual screenings, computational approaches, or just high throughput screening, as you see here from 1,500 to 17, the rate of success is around 1%. So this is really was a very surprising and very heartful result that we had, and gave us a big hope in how to leverage these different algorithms, mainly the deep learning algorithms, towards discovering new molecules against infectious diseases of poverty. This work also have been published in 2021. We have been conducting similar things on using the data set on leishmaniasis. We have experimental validation that is running into our lab in the Institut Pasteur of Tunis. We don't have any results yet, but in parallel, we have been exploring what other people have been doing. And we built up a work on the DeepDR pipeline, which is a network-based approach where they fused information on drugs and diseases, interaction between drugs, and interaction between diseases and drugs into networks, and they were able to predict novel application for already approved drugs against new diseases. So we took a set of molecules that, with a given thresholds, that have been predicted by these people to be targeting leishmaniasis, and we tested them in the lab. So the AI is not our work, but the validation is what we have been doing. We were able to test 40 molecules. Out of them, 4 will have a validated effect against the parasite growth, and 2 of them have not been described for their anti-leishmania effects yet. So these are novel molecules that have been validated in our lab. So this is to demonstrate how we collaborate. So this is a really very elegant way of really using AI in very concrete applications. We are a team of people that have different backgrounds and complementary expertises. And now we are heading to implement this BIND project that is financed by the African Academies of Science, by the European Union and the African Union funds. And we will be building capacity in AI as we are part of one of the very rare teams that are using AI in drug discovery in Tunisia. And the idea is continue the computer-aided drug discovery using bioinformatics, but also developing novel artificial intelligence approaches for drug discovery against different diseases and to tackle issues of interest to the country and to Africa. So that would be all for me, and I would like to acknowledge all the people that have been involved in this work and for listening. Thank you.
I can? Yeah. First of all, I'd like to congratulate you for this very interesting presentation. And let me tell you that a few years ago, when I was in managing the PhD in science at the Medical School of Tunis, I asked every student who was here to register in the PhD, asking him, please go in bioinformatics. But unfortunately, no one of my students accepted to do this. So when I'm in presence of a Tunisian, student and even a Tunisian worker in this field of bioinformatics, I'm very, very, very glad. So thank you very much for this presentation. And I think that it's very important, in fact, to develop this kind of research in even in, as you said, in poor infectious diseases and even in rare disorders that are so numerous. There are not very, there is no a big number of each disease, but they are very numerous, and it's really a problem in Tunisia, in North Africa, in Africa, and the other the other, positive point that you are working now with AAS and African Union, because as you said, for malaria, for leishmaniasis, and other diseases that are prevalent more in Africa than outside. And if you do a good job, I think even big pharma will be involved in the field, and they will really accept to support your work. Thank you very much, Emna.
Thank you for your presentation. You said in your presentation you used the data augmentation technique to augment the size of data set. It helped the model to be more perform and effectiveness. When the model trained in large scale of data, I want to know the methods of data augmentation used in your approach.
Okay, so thank you for the question. The data augmentation is very popular and quite useful in different case studies in AI. But what we saw is that augmenting the data, especially here, so how we augment the data, the existing techniques would use, so I didn't spoke of it. The data, the structure of the molecule is encoded in our database as SMILES, which is just linear representation, textual representation of the structure that can then be read to constitute the structure and then be encoded into a binary vector. And the way of augmenting data that are into a SMILES format is to re-enumerate different SMILES that will basically, it is not basically, they will code for the exact same structure. Okay, so we did different benchmarking, making sure that data augmentation, whether data augmentation is offering better performances or not. And we saw that, as I thought from the beginning, it is like generating artifacts and it is like duplicating data rather than augmenting the data. So for this specific application, data augmentation is not of a big use. This is why we were looking at heterogeneous data sets. So one of the approaches is not to augment data in a very classical way, but it is more to see how, if we introduce or enrich the data set from other sources, how things would go. So it's another way of augmenting the data without having data that comes in the same format or same sources all the time. So it does not work.
Thank you very much for this excellent presentation. I really, I would congratulate you as well. So thank you again for saying that for every model, it's not always the accuracy that matters, and especially for biomedical domain. So since I'm not from the biomedical domain, I'm really interested on the AI and all its applications. I saw in one slide that the feature extraction, we know that deep learning models, they extract the features, and you always use and you also use the traditional machine learning feature extraction. Was it the hypothesis, was it's to enhance the performance, or why didn't you just use the extracted features from DL? And was the convolutional graph, is it apart from how we can say the traditional feature extraction, or just pre-processing step?
Okay, so we have we don't use the feature extractors that we use to feed to machine learning for the deep learning. So these are like two different pipelines. And one of the questions that rises, at which point what we are extracting are influence of the outcome? Is it the algorithm or is it the features? But it is complicated to feed just the binary vectors to the neural networks. And what we use actually is we use graph convolution based feature extractors that will then be used as input for the deep neural networks.
Okay, so this traditional feature extraction using convolutional, which traditionally belongs to deep learning, and then we add it to the extracted features from the deep learning process.
So we didn't do that for this specific project, but we did it for another project where we were comparing, these were image processing.
Okay. And
we saw that features extracted by the deep learning architectures, it was VGG16. So we used the VGG16 extracted features to be fed to machine learning algorithm, regression, linear regression, and random forest. And we saw that it enhances
exactly.
So yes,
exactly. So thank you for also for not using just the deep learning features and enhancing them with traditional machine learning features, and proving again that this enhancement need to be a great field of research, not only in the biomedical.
Well, everyone would say that deep learning is a black box, but still
but we can enhance it.
It is kind of a black box, but it is very important at some point to set up like a pause and say, what are these networks really extracting, especially if you are going or heading to a very concrete application, especially in the biomedical field. So yes, a lot of people are now trying to understand how things, this is why in this particular application, we know exactly what are the features that are extracted. But in other application of the biomedical field, sometimes we don't know, but this is a source of knowledge. It's not about just knowing what the AI is learning. We need to know what are they learning, but because this is in itself a knowledge.
Oh, thank you very much.
Valuable.
Thank you again.
Écoutez, c'est tout est clair. Thank you very much, Emna, for this nice talk. And j'invite tout le monde, j'irai à descendre en bas pour le déjeuner à table. Et j'espère vous voir à temps, à l'heure. J'irai vers 14 h pour le talk de notre cher compatriote Nawel, qui va parler aussi, je dirais, de la santé et de l'intelligence artificielle et du patient. Et de la recherche clinique peut-être. Moi, je ne suis pas un expert dedans, mais je pense que ça doit être ça. Trial, c'est ça? Et voilà. Les essais cliniques, pardon. Voilà. Je suis en face, j'ai une grande spécialiste. Donc, bon déjeuner et puis on reprend à table, au buffet.